Tēnā koutou katoa, ko Ingarani me Piripini te whakapapa mai, ko Lester me Bristol te whenua tupu, ko Tamaki Makaurau te kāinga he Industry Research Liaison o New Zealand Food Safety Science and Research Centre, ko Kyla Archer toku ingoa, tēnā koutou katoa. Welcome to the March edition of the Food Science Seminar Spotlight from the New Zealand Food Safety Science and Research Centre. Thank you for signing up on such short notice. We've had a busy couple of weeks at the centre. And for those of you that we were able to see last week at our Futures Forum in AI, thank you for coming along. Today we have Dr. Olga Pantos from ESR, who will be telling us about the Aotearoa Impacts and Mitigation of Microplastic AIM2 project. The project brings together a transdisciplinary team of scientists, including polymer chemists, microbiologists, and social scientists. And the project's aims include improving knowledge and understanding of the levels and types of microplastics in Aotearoa's marine, freshwater, and terrestrial environments, determining their potential impacts on organisms and ecosystems, and exploring ways to reduce the overall impacts in plastics. Dr. Pantos is a marine biologist with 20 years research experience, specializing in the impacts of human activity on coastal marine ecosystems. Originally from the UK, like me, where she completed her PhD on the microbial ecology of coral disease. She embarked on a journey that brought her to Aotearoa, New Zealand via the USA and Australia. After moving to Christchurch in 2015, she took up a position at ESR, where she's the co-lead of the NB Endeavour-funded Aotearoa Impacts and Mitigation of Microplastics project. So thank you for your time this afternoon, Olga. We have some time for questions after the presentation. So if you do have anything to ask, please use the Q&A box at the bottom. So for now, Olga, I'll hand over to you.
Thank you. Kia ora, everybody. Thank you for joining us this afternoon. So yeah, I want to share with you some of the projects that's coming to the end now. It is a vast project and there is no way I'd manage to fit everything in. So I've just picked a few key things that I'd like to share. Oh, now it's not letting me change. There we go. So yeah, the project team is made up of 20 scientists from 6 institutions across New Zealand, with expertise ranging from microbiology to polymer chemistry and social science. We've had a large number of students as part of the project, which has just been phenomenal. A total of 12 PhD students and 7 master's students. Now, most of these students have finished, but there are just a few of them finishing off. You know, those impacts of COVID have caused delays in people's projects. And so even though we're supposed to be finishing very soon, the students are just starting to get things wrapped up now. So I have some of their results, or some of their projects to share, but not all the results because they're still going with them. So the project was broken into 3 main objectives. The first was to understand the distribution of microplastics in New Zealand. How much was there? What types were there? Where were they? And where were they originating from, and where were they ending up? Then we wanted to understand the environmental impacts specific to New Zealand, and what were the direct impacts, indirect impacts, and things like the impacts on microbial communities, which affect the ecosystem function. Did they interact with invasive species or pathogens? So especially in the marine environment, posing a risk there. And we didn't want it to be all bad news story. So we also wanted to look at some positives and look at how we could help reduce those impacts by reducing the plastic waste going in. So we're going from either end of the spectrum, from, you know, the plastics are only there because of us. All plastic is formed by humans. So how can we reduce the plastic from entering the environment? And at the other end of the spectrum, were there potential microbial solutions, microbial communities that had the ability to degrade the plastics? So this contributed to other work being carried out around the world. So before we start trying to work out what the risks from plastics are, we really need to know what is plastic. So we're surrounded by plastic in our everyday lives, everything from our computers, our cars, our clothes, our food, everything is surrounded by plastic. So people just sort of think of plastic as plastic, but plastic is very much more than that. The plastic is a combination of the synthetic polymer, so your polyethylene or polypropylene, but then it's got a large number of additives within the plastic matrix as well that helps give that plastic item its function and form. And it's also things are added in when the plastics are made to enable them to make them. So things like thermal stabilizers, so when they're melting them, they don't start to degrade. Your colorants, your biocides, UV stabilizers. You see a lot of plastic products meant for outside saying that they've got UV stabilizers on them. And if you have recycled plastics, you can end up with a lot of other things in there, intentionally added substances, which are associated with the plastics that have been recycled, but there may be residues of what the previous plastic item was used for, and these will include pharmaceuticals, other care products, chemicals. So they may all be within your recycled plastic as well. So there's actually no way of knowing what's within those plastics because there's no requirement to identify any of those other chemicals. So what you'll see on the packaging is what polymer it is, which really doesn't give too much away. So these were just some of the examples of those different intentionally added chemicals that I've mentioned just before. So these chemicals are produced in high quantities, and you can get some added to the plastics in really high levels. Things like phthalates for plasticizing agents, they can be 40% of your mass. So you've got large amounts there. And these additives are not bound within the plastic, so they can migrate out as well. Many of them are toxic. There's very little understanding of many of the chemicals that are added into plastics on their actual risk to the environment. And so there's huge amounts of work being done at the moment trying to understand this. So we know what plastics are. So what are we talking about when we talk about microplastics? So the definition of a microplastic is a fragment of plastic or a particle of plastic 5 millimeters and smaller. The lower size limits varies a little bit. There are about 3 different lower definitions, but one that's been settled more on at the moment is 1 micron, and this is being used within the UN's plastics treaty as well. So that's just the size range I've listed on here. So microplastics are broken down into primary microplastics and secondary microplastics. So your primary microplastics are those plastics particles that are made to that specific size, to a size that's less than 5 millimeters. So these will include your microbeads, your glitter, furry nail polish. If nobody's heard of it before, apparently it's a thing, and it's tiny little microfibers that you put nail polish on, then dip your nails and you end up with furry nails. I mean, there's it's limitless the types of primary microplastics you can get. So secondary microplastics are the breakdown products of larger plastic items. So fibers from your synthetic clothing or furnishings. You have synthetic carpet, it wears out, those fibers are released. Solid plastics that get broken and crushed, and they crack and they become weathered and they gradually break down. And once they get to that microplastic size, they just continue breaking down and eventually become nanoplastics. So the environmental impacts are quite wide-ranging, and we certainly don't know all of them yet. But there are different properties of the plastic that can affect how they interact with the environment. So these include the polymer itself, where it's polyethylene, polypropylene, polyester, and they'll have their own inherent associated chemicals with them, and they may have other ones added in. So you've got those combinations. The size of the particle can affect how it interacts as well with organisms, and the morphology. So you might have a fiber, a fragment, or a bead. Fibers and fragments cause more of an immune response, just physically, because they're a bit pointy and sharp, whereas beads are smoother and may not have a similar effect. The microbial communities that may associate with plastics may be different. That may be based on the type of polymer or the associated chemicals. The age of the plastic affects its surface texture, which affects the take-up of environmental chemicals onto them and the microbes that are associated with them. And then, of course, depends on what organism it's interacting with as well, or the life stage of an organism it's interacting with. And then there are, I'm sure, other factors that we've not identified yet that may be altering how they interact with the environment. So some of these physical effects include gut blockages, nutritional reduction because of that and starvation, inflammation and infection, physical damage and reduced motility, reduced biomass. So if you're not getting as much food, your growth efficiencies affected, reducing productivity, and these can have knock-on effects to reproductive impacts as well. So plastics will act as a good vector for contaminants, including those already in them or that they picked up from the environment. So they can help increase the uptake of contaminants to an organism. There's evidence of it's affecting the bioaccumulation and trophic transfer of these chemicals. And many of these chemicals have already been identified as endocrine disruptors, which again can have a reproductive impact. So the effects can be indirect through the microbial communities that may associate with the plastics, altering the biofilm formation or microbiome of an organism. This can affect the organism itself or affect ecosystem functioning, so nitrogen cycling, oxygen production. In the marine system, biosecurity is a concern because of things settling on. In the bottom right-hand corner of the screen, there's a expanded styrofoam ball which has a baby barnacle settled on it. This was collected through surface water trawls off Napier. And it may be a local barnacle or it may not be, but you can see that they can pose a risk for transporting things further than they may do because plastic's so lightweight, very buoyant and very resilient as well to degradation. So it can keep going for much longer through currents that natural materials may not. I mentioned a bit later one of the experiments we did looking at plastics in wastewater treatment plants because the microbes can settle on them and act as vectors for potential human pathogens out of wastewater treatment plants because they can evade the treatments. And then there's increased horizontal gene transfer and virulence and increased AMR as well. So with the distribution of plastics in different environments that may not otherwise happen, it can increase those risks as well. Our exposure to plastics has long been just considered through food. And it can be fresh foods or processed foods. You have fresh lettuces, vegetables can collect airborne microplastics. So there's evidence of that. There's transfer as you eat the food, even with some washing, unless it's very, very thorough, you'll still have particles there. Processed foods, plastics coming from the machinery, the equipment, and of course the plastic packaging as well. Even including when the food is within the packaging, there's evidence that for some items such as rice, scratchy things inside plastic packaging, they actually grate slightly the inside of the bag, creating more plastics in there. Inhalation, that is a direct route, large numbers of plastic particles in the air that we're breathing in every day. And so we need to be very cognizant of this. So this may also be falling onto your food at your dinner table as well and ingesting it that way. And there is evidence of dermal uptake as well through damaged skin. So the area of research around human exposure and impact is relatively in its infancy. There have been a lot more studies that are starting to publish just at the moment. So we'll see those coming out soon. But so far, microplastics have been identified in lungs, lung tissue, in blood, in placenta. There's another, I think there's a second study out on placenta and feces as well. Some of the impacts identified so far include inflammation of the gut and damage to lung cells. And this damage seems to be dependent on the polymer of that fragment as well. And a very recent study, I think reported last week, was a link between heart failure or heart attack and levels of microplastics within the blood as well. And they're causing some micro blockages in the blood vessels. And a higher level impact to humans is on food security. So these impacts that the plastics can have on the growing environments can affect, and we're talking large and small plastics, can affect the safe and production of food to levels that are required. Right, okay. So food production is one of the major users of plastic, all the way from the field to the supermarket shelves. This was the first study to look at plastics, microplastics in New Zealand soils. And this work's being carried out by Helena Ruffel as part of her PhD project. And she's just coming to the end now, busy writing papers and her thesis. So I don't have any results of hers just at the moment, but those should be being published soon. But in food production, you've got the netting, the clips, the mulch, the compost, large, so green domestic waste compost contains a large amounts of plastics. Got the example of the plastic food stickers, but we find an awful lot more in there than just those. There's an example of the plastic mulch used with some with a crop here in New Zealand, and that mulch, even if it's gathered in, is going to be releasing plastics to the soils as it's laid out, and then when it's gathered back in. And in picture B, there's a fragment of that mulch that was found after pulling up the mulch from the previous year's crop. Compost is often applied to horticultural soils as a natural fertilizer, but from Helena's work, we're seeing a large amount of plastics in compost and in vermicompost, and all of this is transferring into the soils. So these are just a few examples of microplastic fragments that we identified from these soils. So we've got PET, or it would be a polyester fibre fragment of PVC, 2 fragments of PVC. And these were taken from soils just in the same area. I think I've mentioned these ones already before. So another thing to be considering is that we're not only creating a potential route of microplastics into our food, but we're also going to be impacting the soil ecosystems themselves. which could then have an effect on the quality and quantity of the food that can be grown. So marine activities, I'm sure you're all aware, use a large amount of plastic. It's such a wonderful material to be used out at sea. It's resilient, it's cheap, it's buoyant, and you can use it for so many things, and it can be modified with the additives to make it very resilient with the UV stabilizers and all kind of other additives that you can add in there. So I just wanted to go through a few of the studies we've done. Like I say, over 5 years with so many people working on it, there's an awful lot of work that's been done. And I just wouldn't do it justice to try and squeeze it all in now. So there are just a few things I wanted to highlight. So some of the work we carried out was looking at beach sand in 3 regions across New Zealand. We looked in Northland, Waikato and Canterbury. So the map here is the example of Northland. All of the analysis of the sand samples were done by our SCIAN team members, and they developed a new method to optimise the isolation and identification of small particles, which are standard methods up till now have been neglecting, just because it's quite difficult to do and time consuming. So the majority of the methods used to date have focused on particles that are several hundred microns. And so they've been missing the smaller size fraction. So people at SCIAN managed to develop a new method which optimized for large and small. So these graphs just show the proportion of different polymers that were found within the large size fraction and the small size fraction. And so there was no real surprises there. Your usual suspects of those high production polymers, polyethylene, polyester, polypropylene. What was very interesting and more worrying is if we look at the size fraction distribution. So this is for all 3 regions, and you can see consistently that the majority, by several orders of magnitude, were in that size fraction of less than 25 microns. So if we'd been carrying out this work using the older methods, we may have been only identifying a handful of particles per kilogram of sand. And we'd be feeling confident that our coastal environments were pretty much free of plastic pollution. However, with this new method that was developed, we can see that there are several thousand particles per kilo of sand. And who knows what's below, and those even smaller size fractions. So the concerning thing about this is the sheer number of particles of that very small size range, which the researchers in human health are concerned about, because they're at that size where they can start to cross membranes and cause a whole range of other issues, because as things get smaller, the impacts vary or change as well. So being able to identify that there's still plastic there, just much smaller, is really valuable as we go forward, and we want to be able to improve the situation and make change. It will enable us to understand the current state, where we do need, where there are problems, what the actual level of risk is in an area. Okay. So we've also looked at shellfish. It's just an example of some of the mussels that we've looked at, and again, there's no real surprises, just like any other studies of mussels done in New Zealand already, and even New Zealand mussels that have been brought overseas and tested seeing similar things. A range of sizes, same polymers that you'd expect to see, those high production ones, the polypropylenes and polyethylenes. So it wasn't a surprise when we saw the same. We've also looked in some infaunal species, so the ones that live in the sand that are collected more as recreational, and we're seeing the different selection of polymers. And this may be partly due to the changes in buoyancy of the different polymers. And also we saw differences between farmed mussels and wild caught mussels, because the farmed ones are on the ropes and the wild ones are on the rocky benthos. Right. So this was another part of a student's project, and this was carried out by Hayden Masterton as part of his master's project with the University of Canterbury. He had several focuses within his project, and this was one small part of it. So Hayden worked with the team at Litter Intelligence. This project is the Litter Intelligence project is run by Sustainable Coastlines. They're based in Auckland, but every 3 months they go out with a large group of volunteers to a large number of locations around the country every 3 months, and they do the exact same area every 3 months, and they collect all the plastic and they identify everything, and it's all on their database, which is available publicly online. It's a fabulous resource. They're doing some incredible work. So Hayden went with them to some of their locations and took aside a subset of the samples they were finding, and we collected from 21 study sites and took away 957 pieces of plastic. So interestingly, the two worst sites for high levels of plastic were Whangarei and Invercargill. So the average density of plastic in area was 0.099 items per square metre. All of the pieces of plastic were collected with nitrile gloves and placed in foil so that they could then be analysed for trace element analysis and organic contaminant analysis. So Hayden did the trace element analysis, and Jamie Bridson from Scion did the organic contaminant analysis. So he did that as part of his PhD project, which sat under AIM2 as well. But I don't have time or knowledge enough in chemistry to really talk about his work. But I'm sure if you wanted to get in touch, he'd be happy to share. So they got their pieces of plastic, and they were limited in size by needing a piece that was big enough for them to be able to do the analysis. So there were a lot of fireworks casings, lollipop sticks, food and drink packaging, fishing lures, lines, ropes, and then lots of fragments that they weren't able to identify their original origin. They compared all the polymer types, looked to see what the polymer types were. And again, it's those high production plastics that were the dominant ones, polypropylene, polyethylene. And they broke them down as well, classified them by the colour, because of course the colour is determined by the additives, or they use additives to create the colour that's wanted. And that can affect what the chemicals are within them. So it's just a summary of the trace elements that Hayden was finding there. A few things of interest. These two in very high concentrations. They're known to be used as fillers, and these four here are commonly used as pigments in the plastics. So we have these plastics, and well, how much harm can they do? So Hayden wanted to have a look and determine this. So he took two plastic items, quite quite different in use. So there was the blacks are the firework casings, and the whitey green is the lollipop sticks. So to determine the bioavailability and potential risk to organisms that come into contact with them or ingest them, more specifically, he simulated gastric fluids and simulated a digestion if they'd been eaten. So to understand the effect of them also being out in the environment for a long time, he was able to use brand new lollipop sticks, compare them to the ones he collected on the beach. But obviously for health and safety reasons, he was unable to get fireworks and take them apart and use unused fireworks casings. So in the image, hopefully you can see that that lollipop stick that had been floating around in the environment for a long time, it's very cracked and it's got green algae growing in those surfaces. So as they age, the surface area becomes greater, and so more chemicals can, or there's more interaction between microbes and chemicals in the environment as well. So when he had done the digest, and he then examined or tested the fluid that they'd been in, and there was quite a bit of difference. So I won't go into details on this, but you can see that the fireworks had higher concentrations and more elements present, but it was the lollipop sticks that had the greater level of bioaccessibility. And between the vertebrate and the invertebrate, it was the vertebrate that had the greater. So when this analysis was done, one thing that didn't have time to do was actually go back and redo this using a gastric fluid replication for seabirds, which would have had a higher level of oils in the stomachs, just based on their diet as well. So this isn't that representative of seabirds, but be very interesting to be able to go back and look at that as well. Yeah. So I feel like I've flown through this and I could have told you lots more, but yeah, one thing that's feels really important when we're talking about plastic contamination and the risks is that the risk to the environment and to us is not only at that point of use at the end, and we should be considering the whole life cycle of the plastics. You know, where is it coming from? Where is it produced? From the point it's drawn out of the ground as oil, then turned into plastic, then transported and made into something else and transported again. Every stage of that is a potential point at which it can become pollution in the environment. And of course, plastics being very buoyant, many of them, some of them will sink. There's the potential for them to travel large distances and to not only affect the areas where they turned into pollution in the first place. So I like this example of it was a container ship that lost a container containing HB inkjet cartridges back in 2014 off the coast of New York. In 2021, those cartridges washed up in the Arctic, and they were able to compare with where the currents were going as well. So there, see one of those cartridges. So not only have you got the risk from the polypropylene casing, you've got all the additives that are in them. And with this being something that's like our computers, they've come in contact with electronics. There's usually flame retardants as well in there. Lots of things I never even considered about until doing this project. So you've got the chemical risk from that, and as it breaks down, they'll turn into microplastics and carry that risk and different risks with them. But there's a clear example of that biosecurity risk that plastics out in the environment can cause as well. And with the biosecurity risk as well, you have the risk of pathogens as well. And I appear to have lost a number of slides, which I'm sorry about. Wanted to talk to you about the, we put plastics out into the wastewater treatment plants. At the same time, we put them into the marine system. But I just wanted to explain, to tell you about some that we put in the wastewater treatment plant to look at the microbes that were became associated with the different plastics and plastics that were aged and unaged. So this was work that has been led by several students who are under the supervision of Gavin Lear at the University of Auckland. They are just starting to write up now. So I don't have their results to be able to share at the moment. Very soon they're all being put together in papers. But we were seeing differences in the human pathogens and pathogens of interest in the UN database as well, that were present on the plastics over different time periods. And there carries the potential for it to make its way out into the environment. And also, if we look into the biosolids, which may go into vermicompost, which may then end up on land. So there are huge cycles of this plastic that seem to be able to make their way one way or another out into the environment, into our growing environments as well, either in the sea or on land. I'm sorry, I don't know what's happened to those slides. But if anybody had more specific questions about what we did, I'm happy to share. So there is just too many people to thank who have helped with this project. It's been over 5 years plus a little bit because of COVID. There was a 6-month extension, and we couldn't have done it without all the support and all the hard work from people. So I didn't want to list anybody in case I missed people, which I'm sure I would have done. So thank you very much. And any questions?
Great. Thank you so much, Olga. That was it was really interesting to hear about all the work that that you've been doing over the past 5.5 years. I've not had any questions come through, so yeah, this is your invitation now if you wanted to add anything. I had a question. So you know, you come to the end of this 5-year project. You did say at the beginning it wasn't all doom and gloom, but so what are the next stages for this research, and you know, for potentially with public engagement or with industry engagement?
Yeah. Well, I guess the thing is we know it's a problem, and there is no. We need to keep a track of it and keep looking at all the other potential risks. But we know it's a problem, and we really need to do something about it. The biggest thing we need to do is stop the production of plastic pollution, not stop the production of plastic because it does have very many good uses, but we need better plastics, and plastics that are more in tune with the environment and don't pose those risks. And whether this is a plastic that you can use in your fields, which does not shed, which is super durable, but still does its job, or it's one that if bits do break off, they will naturally degrade and not cause any harm. And so there's unintended consequences. So we are hoping to shift our focus to looking at some of those, you know, finding those alternatives, finding more environmentally friendly materials for both horticulture and aquaculture. So we're working alongside industry to find things that work for them and don't have any unintended consequences and regrettable substitutions, which there are plenty of examples out there where something has been a problem and someone has come up with a solution without actually testing that it is a true solution and good in all respects. Yeah, so that's what we'd like to be able to do now, but that requires more funding. I guess I should say it's on the slide, but say again, this is all made possible by our MB funding for the Endeavour project, and a fantastic team. So this is where we want to go. Stop looking at the bad stuff and try and find some solutions.
Great, thanks. We've had a question come in from Julia, and kind of putting you on the spot without you being a polymer chemist, but she's interested in your thoughts on how likely it is that some of the microorganisms will eventually evolve to degrade some of these different plastics into safe components.
Yeah, there's an awful lot of studies out there that are claiming to have identified microbes that are able to degrade plastic. So actually, there's a database that one of our students, supervised by Gavin Lear at the University of Auckland, he created a database where you can look up your different polymers, different plastic polymers, and find all the species that have been, and enzymes that have been identified as having the ability to break down plastics. And then it will take you to all of the publications. However, when you go, so this is absolutely fantastic tool that Victor has created, and it's online and free. But when you start gathering together all that information, people have been using different definitions of what's the plastic having been degraded. So if you have additives of very high concentrations within your plastic, they may be things that the microbes can eat easily. So if they do, you have a large loss in mass. So it appears like your plastic is being eaten, but actually it's the easier to eat things within that polymer. So your plastic is still there and has the potential to break down into microplastics. So there's an awful lot of that out there. And for those things that are truly metabolizing or producing enzymes that can degrade the polymers, they're clearly doing it very, very slowly, or we wouldn't be in the mess we're in. So I'd like to hope that there is potential, and I know several groups that we're collaborating with who are working on trying to achieve the same goal and find those microbes out there that have that potential, and hopefully harness it or use the information to make better plastics that are more easily broken down you'd hope under controlled circumstances, because you don't want to suddenly find the things around you every day falling apart. That could be quite difficult.
Yeah, no, you don't want all your muscles growing on ropes to suddenly just be dispersed again. It's the whole point of putting them on lines. Oh, Kerry, I'm just going to come to your question quickly. Yes, we'll record this. Well, we have recorded this, and it will be put online shortly. I've got another question from Tim. Had great talk and data. Thank you, Olga. Is there the intent to create some easily digestible resources or media pieces for the general public to raise awareness of issues and things that people can do to help?
Specifically from the project?
Yeah, specifically from the project.
During the life of the project, working with Paracore, they've been one of our partners. They've put together a number of resources, and also our partners in the FO, Eco Matters and Te Kauramaki. There are a number of resources as well that was developed with them to share knowledge and understanding through the communities. So we've not done anything that's been broad scale to everybody, just working with those groups with our local communities. Hopefully if we get more funding, it certainly is a key area of the project will be to ensure that we can do that sort of thing, because that's where the real change is going to happen.
Yeah. Great, thank you so much. I think we're out of questions unless anybody's got a last minute quick one. But so yeah, I guess I'll wrap up. Thank you so much for your time today, Olga, and for being able to put together this presentation. Yeah, I said I'd seen some of the early things when you'd initially applied for and received all the funding when I was based at the University of Auckland. So it's really great to see what's been done over that time, and really great to hear of work in New Zealand that is working on such a global concern, and hopefully the work that you've done can be a big, a leader in global efforts to reduce microplastic pollution. So yes, this has been recorded and will be on our website later this week, along with all our previous seminars. So you'll find it on our homepage under events and seminars on the top right hand side. Our next session will be advertised to all of you in a few weeks, so keep an eye out, and we hope to see you next time. So have a great rest of your week. You've got 15 minutes back. Ka kite ana.